Hello everyone, my name is Irfan Khan and I am an honorary research scholar with the Department of Chemistry at the University at Albany, New York. Today we are beginning our spring molecular and cell biology lecture series where we will explore key concepts, techniques and advancement in this exciting field. I look forward to engaging in discussion and hope this series will deepen our understanding and more key concept of molecular and cellular processes. Let's get started. So our first topic is cell culture theory and practice. So before start, there are some things that need to be remembered. First of all is what is scientific research. So uh, uh, we will learn definition of scientific research and steps involved to perform scientific research. So systematic investigation to discover new knowledge or validated existing theories through observation, experimentation, and analysis involved in scientific research. Key steps include defining the problem, formulating a hypothesis, conducting experiments, analyzing data, and drawing conclusions. Most important thing, keeping proper laboratory notebook is a detailed chronological record of experiment observations and procedures, ensuring reproducibility, accuracy, and legal validity of research. And also, most important point is review of commonly used lab math. So to perform lab math, this is essential mathematical calculations used in laboratories such as dilutions, molarity, and unit conversion, ensuring precise measurement and reliable results. Okay, so what is cell culture? Cell culture is removal of cells from an animal or plant and their subsequent growth in a favorable artificial environment. So artificial environment such as cell culture medium in a form of liquids uh, into the glass tubes or in petri dish or in a flask. Uh, what is primary culture? Culture after the cells are isolated from the tissue and proliferated under the appropriate conditions. Subculture after first initial culture passage by transferring them to a new vessel with fresh growth medium. After first passage, primary culture becomes known as cell line, and normal cells usually divide only a limited number of times. So this is finite. So because those normal cells they have a limited life cycle, so after a couple of passages they will start dying. So what are continuous cell lines? Cell becomes immortalized or uh, prone to death and can proliferate indefinitely as long as culture conditions are favorable. How we achieve continuous cell lines? There are a couple of methods routinely used in cell culture studies. Most famous is transformation using plasmid or some other nucleic acid material, spontaneous by time, using some chemical inducers, and a viral infection. So we will discuss those methods separately in other classes. Currently, majority of commercially available cell lines are immortalized and derived from human cancers. So one volunteer lady, Henrietta Lacks, she kindly agreed to be volunteer to provide her cervical cancer tissues to be used in research, and they are currently known as HeLa cell lines, and they are most widely used cell lines in molecular and cellular biology. So how cells differentiate? So usually on the basis of cell morphology, There are three basic categories based on shape and appearance. First is fibroblasts, like elongated shaped adherent cell lines. Adherent means they are stuck on the surface of the petri dish or cell culture flask. Epithelial, like here on top of panel, so they are polygonal and adherent cell lines. And the lymphoblast, like spherical, and they usually grow in suspension. They don't like to grow on surface. And here is the example of fibroblasts, like those are usually spindle-shaped cells. So what are applications of cell culture? Studying the normal physiology and biochemistry of cells, metabolic studies, cellular pathways, the effects of drugs and toxic compounds on the cells. In biotechnology and biopharmaceutical industry, so those cells are usually very handful to test new drug formulations, and these are also favorable to study mutagenesis and carcinogenesis. As I mentioned earlier, cell lines are used for drug screening and drug development. And in the industrial environment, large-scale manufacturing of biological compounds, especially vaccines and therapeutics proteins, usually synthesized using cell culture systems. So there are a couple of advantages and disadvantages of using cell culture systems. Uh, for advantages, we can see control of the growth environment. We can control how much nutrients we can provide in medium, what type of condition, like oxygen, carbon dioxide, temperature, everything is controllable. And characteristics of cells can be maintained over several generations. Good reproducibility between experiments. Cultures can be exposed to reagents, drugs at defined concentrations. Avoidance of legal, moral, and ethical problems of animal experimentation. Beside advantages, there are a couple of disadvantages as well. Need standardized techniques and culture conditions to ensure reproducibility. Aseptic technique is critical. Cells can change after successive passages. Immortalized cells are not quite easily handleable. Now we will see what are culture conditions. So a substrate needed, that is called cell culture medium, that provide essential nutrients in form of glucose, salts, and some growth hormones, growth factors, gases, especially oxygen, regulated environment in terms of pH and temperature. So culture conditions are varied by cell lines. So ETCC is a big provider of cell culture material using in research and biopharmaceutical industry. So let's say example TSP1 cell line. This is acute monoballistic leukemia cell line. So manufacturer provided this instruction what type of medium you need to use. So this is ATCC formulated RPMI 1640 medium. This is also available through various other vendors. And they mention we need to add uh, beta mercaptoethanol to a final concentration of 0.5 millimolar, and fetal buoyant serum or FPS at final concentration of 10%. Beside this, they also provide its subculturing protocol, cryopreservation, and cultural conditions. So we have to strictly adhere to those conditions, otherwise our cell will not grow or will start dying. As we seen in earlier slide, what is media? So this is a substrate, and it provide essential nutrients, growth factors, gases, especially oxygen, and regulated environment, pH and temperature. To grow cells in laboratory, we need a couple of equipments. First of all, we need biosafety cabinet, specialized hood that facilitate the inner air clean and prevent outer air to enter into the hood. We need incubator, water bath, centrifuge, refrigerator, microscope inverted especially, hemocytometer for counting of cells, cell culture vessels, pipettes, micropipettes, and waste containers. In list of equipment, first is biosafety cabinet. So what is biosafety cabinet? Enclosed ventilated laboratory workplace for safely working with materials contaminated with or potentially contaminated with materials requiring a defined biosafety level. The biosafety cabinet is also called laminar flow hood, maintaining a constant and unidirectional flow of HEPA filtered air over the work area. And this airflow is horizontal, blowing parallel to the work surface, and also vertical, blowing from the top of the cabinet onto the work surface. Provide protection to user and the culture. Another variety of biosafety cabinet is the class 2 biological safety cabinet, and it has three key features: a front access opening with carefully maintained inward airflow. On top, HEPA filtered vertical unidirectional airflow with the work area. Another HEPA filter exhaust air to the room or exhaust to a facility's exhaust system. Type A to 70% of the HEPA filter air is recirculated, while 30% passes through an exhaust HEPA filter and is discharged into the lab. Now, how to operate biosafety cabinet? So, raise sash, that is glass door, to the appropriate level. No alarm sounds, and turn the blower on automatically. There are marks on both sides of left and right on the glass holder. So when you reach those marks, if you will go beyond those marks, so equipment will start beeping. And when you lift the sash up, blower automatically turn on. Let the blower run for 5 minutes after turning on to purge the air. Don't directly start using your work or experiment because you need to provide clean air. Load all necessary supplies prior to working in the hood. So going again back and to forth to bring pipette box, pipettes or culture medium or tubes or flasks will cause contamination. Spray down items with disinfectant, especially 70% ethanol, before loading into the hood and after they are removed from the hood. Clean up any spills promptly because cell culture medium especially is most favorable uh, food for bacteria and other growing contaminants like mold. So to separate clean and dirty slides in the areas, pipetter in the front right where it can be reached easily if you are right-handed. If you are left-handed, you can put on your left hand. Do not overload the cabinet. Keep tidy. Work in the center of your work area, not on one side. Do not block the rear or front grills. That will cause obstructing airflow. Minimize disruption of airflow turbulence. When finished, lower the sash, turn off the blower and turn on the UV light at least for 15 minutes for a sterilization purpose. So you can watch YouTube video for function of biosafety cabinet. Next, we will learn about incubators. So incubators provide appropriate environment based on cell type. Mammalian cells usually grow at 37 degrees centigrade. Insect cells at 22 degrees centigrade. They are made up of stainless steel for cleaning and decontamination. Water pan to provide humidity and prevent evaporation. Attach CO2 tank or CO2 supply 5% to keep cells at correct pH, and also prevent from physical hazard. Physical hazard means excessive temperature or atmospheric air or too much humidity. Those are all harmful for the cells. Other equipment are including centrifuges, microcentrifuge that hold microcentrifuge tubes, tabletop centrifuge, conical tubes 15 to 50 ml, those required for sedimentation of the cells. Sealed rotor refrigeration may be needed for some cell types. Cells are palleted at 150 to 300 G RCF. Any higher can damage cells. RCF calculated using rotor radius. Speed RPM needed to achieve RCF may differ between centrifuges. What is cell culture safety? So, to prevent spills, splashes, aerosols, and accidental punctures. Different BSL depending on the cell line and experiment. BSL1 is mostly used for majority of cell lines. Those are normal type of cell lines, and we are not using any contaminating agent, especially biohazard bacteria and viruses. BSL2 also used for primary cells, purposely infected with pathogens, but those are not highly contagious. In both environment, you will need appropriate PPE, personal protective equipment, gloves, lab coat, disposable, and face shield as needed. To work in cell culture laboratory, we need to follow aseptic techniques. Successful cell culture depends heavily on keeping the cells free from contamination by microorganisms. Common sources of contamination are non-sterile supplies, media and reagents, how to ensure sterility, airborne particles laden with microorganisms, unclean incubators, dirty work surface. In my practice, I usually keep my media and reagents separate. I won't share any of my cell culture medium, FBS, antibiotic, and other additives that I routinely use. If someone need, I provided a separate aliquot from fresh stock. So to maintain a sterile work area, we need to proper disinfect the area with 70% ethanol, simple green, or 10% bleach if we are working with contaminating or contagious viruses like HIV. Also, follow good hygiene, good hand washing before and after working in biosafety cabinet. Now, what is contamination? Right? Contamination can be biological, bacteria, molds, yeast, viruses, and mycoplasma. So, to prevent contamination, we use antibiotics to the media, but still there are chances of cross contamination, reputable sources for cell lines, and can be difficult to detect, especially for ELA cells. In cell culture environment, mycoplasma called a scientist's worst nightmare. They altered the cell properties, skewed research results. Small free-living bacteria undetectable by naked eye or microscopy. Lack of a cell wall, resistance to many common antibiotics, complete with host cells for compete with host cells for biosynthetic precursors and nutrients, and can alter DNA, RNA, and protein synthesis. Research showed impact on the expression of more than 100 generation. Sources in between cell lines from reagents, special detection method required, and anti-mycoplasma reagents also commercially available. So now we will discuss what are cell culture media and their types. So natural media are biological fluids like plasma, serum, amniotic fluid, tissue extract, liver, spleen, bone marrow, and blood clots. Artificially, those are synthesized in commercial environment. So two type of those media are base media minimal essential media, and Dalbico modified essential medium. And complex medium are RPM 1640. Those are required for a special cell type. So cell culture media usually contain amino acids, carbohydrate sources in form of glucose, salts, vitamins, buffering agents to regulate pH, phenol red for color change indicator. By time when cells grow, they release gases and they turn into pH change, and that phenol red is usually turned from red to yellow by time. And some cell type required various concentration of fetal buoyant serum, uh, nutrients, hormones, growth factors, and they can be complex as well. Common type of cell culture medium in use are Eagles minimal essential medium EM, Delbicos modified Eagles medium RPM I1640, Ham's nutrient mix F12 medium, Isobes modified Delbico medium. Common cell lines and their recommended media. So HeLa B is epithelial origin cell line and belongs to human, and it requires MEM plus glutamine plus FBS and non-essential amino acid. So this medium cell line is used for terminal tumorigenicity and viruses studies. HL60 is lymphoblast cell lines and origin is human. Required RPMI 1640 with the glutamine plus FBS, and this is used for differentiation studies. Similarly, there is a list of different cell lines that you need use in research and biotechnology industries. So nowadays, CHO7 cell line, this is epithelial origin, and this is from hamster model. They required HAMS F12 medium with glutamine and FBS. So this is used for nutritional and gene expression studies. Also, HAC293 cells commonly used for uh, transfection of different genetic material, and this is also epithelial cell line and belongs to human, and it requires EM plus glutamine, non-essential amino acid, and 10% FBS. Why we need serum in media? So serum contains nutrients uh, cells need to grow. Growth factors, minerals, lipids, and protein. Most use include bovine calf, fetal bovine, and horse serum. Human serum used for some cell lines. Fetal bovine serum most widely used. So, how to isolate serum or plasma? When we have serum, it is in clot form. So we need to centrifuge at high speed to separate serum from the clot blood material, and those isolated serum aliquots keep at minus 20 degrees centigrade frozen, and by the time of use they will be thawed. And what is the difference in the plasma versus serum? So plasma is not clotted because it was added in anticoagulant. Anticoagulant keep plasma in suspension, blood in suspension. So when you centrifuge, you will see three layers. One is sediment of red blood cells, in between buffy coat that usually have white blood cells, and top layer is plasma. So we are not using plasma to add in cell culture studies, but plasma is still used in some other studies. So there are advantages of serum in media. Good is stimulus cell growth, aids in cell attachment, aids the buffering, binding proteins to certain toxins. There are some bad things. Lot to lot variability, batch testing required, ethical concerns, especially use of animals, risk of contamination, mad cow disease, interface with downstream purification, isolation, and other procedures. So we need to follow certain protocols for cell passage, cell concentration, cell counting using hemocytometer, and cryopreservation. Cell passage, also known as subculture. So this is a replacement of growth media when media turn yellow colored, and regularity depends on cell properties of your cell line. Materials needed differs on type of cells. Suspension cell dilute into fresh media in a new flask. We don't throw all of the suspension cell medium after growth because it has some nutrients that cells still need. Uh, Adherent cells they require dissociation region, in example trypsin and PBS, and also in some cases we need mechanical disruptor. So to save cells, procedure is called cryopreservation. Cells are cryopreserved to avoid loss by contamination, minimize genetic changes, and continue cell lines. Passage number, avoid aging and transformation in finite cell lines. Cryopreservation media generally consists of base medium, cryopreservative 7.5 to 10% DMSO, and a protein source 10% FBSA or FBS. So in this slide, I have attached how to freeze cells and how to thaw cells videos. How to count cells. It is often necessary to determine the concentration of cells in a suspension. A counting chamber can be used to accurately determine the number of cells per unit volume of suspension. Most widely used is the hemocytometer, but nowadays the automatic cell counter available in different research and diagnostic environments. So in a manual cell counting system, we use vital trypan below that bind with the live cells. Can be used during cell counting to calculate cell viability. So live cells with intact cell membrane are not colored by the dye. They will be transparent when you see under inverted microscope. Dye passes through the membranes of the dying dead cells and they become blue, so we can easily distinguish between dyed and live cells. Hemocytometer is a specially made device, and this like a 3 by 3 grid in size. There are 9 large squares resembling a tic-tac-toe grid. So those 9 large squares are 1, 2, 3, 4, 5, 6, 7, 8, 9, and the central square divided into 25 small squares with the a triple lining. 4 corner squares divided into 16 small squares. Procedure for counting cells is mix 10 microliter of cells with 10 microliter of trypan blue, 1 to 2 dilution final, pipette 10 microliter into the wells of one side of the huge chamber. Place hemocytometer on the microscope and bring into the focus using the 10x objective. Enumerate the cells found in the 4 large corner squares. Calculate an average per square. Calculate cell concentration per ml. Cells per ml equals to average count per square into dilution factor if applicable. In our case it was 1 to 2 into 10,000, that is magnification of the our 10x lens. Calculate cell viability. Percentage of viable cell equals to 1 minus number of cells divided by number of total cells into 100. So sample cell count procedure here includes cells in green checks. Those are on the inner side of the large square. those outer side of the large square cells will not be counting. Exclude these cells touching middle line on bottom of the right. So corner square number 1 equals to 42, corner square 2, 37, 40. So average count is 38.5. Now cells per ml equal to average count per square dilution factor into 10,000. So cells per ml was 38.5 into dilution factor of 2 multiplied by 10k. So our cell count was let's say 770,000 or 7.7 to 10 to the power 5. We can count cell viability in our suspension cells. So there is a easy method available, and so we need to stain cells with the trypan below tie, and we can count the below cells. So here like we have 15 cells. So we can easily count cells out of viable cells in provided cell suspension, and here is the calculation that can be used to achieve total number of viable cells. So uh, this is the first lecture of our molecular and cellular biology course. So we'll continue provide more research and teaching material on different topics in the upcoming videos. So feel free to send me questions if you have any in your mind, and talk to you soon again. Have a good day. Bye.